Welcome to this e-module. This program is designed for the students of Bachelor of Science, 3rd year, in the subject of Botany, Semester 6, course code BOC 108. Title of this course is Cytogenetics and Plant Breeding. The title of this unit is Mendelian Genetics and its Extensions. Module name is Gregor Mendel's Experiment with Pea Plant. I am Ms. Emma Fernandez, Assistant Professor in Botany from Tempe College of Arts and Science. In this module, we are going to learn about life of Gregor Mendel, Mendel's experiment on pea plant, and Mendel's observations and result. At the end of this e-module, the student will recall the life of Gregor Mendel, will describe Mendel's experimental procedure on pea plants, and also list down Mendel's observations and results in pea plant experiment. Now let us learn about life of Mendel in brief. Gregor Johann Mendel was the pioneer of classical geneticists. Apart from being a scientist, he was a priest by profession. Mendel finished his theological studies in 1848 from Augustinian monastery at Brunn in Moravia. Later, he was then appointed as a substitute teacher in the Imperial Royan Gymnasium in Znaim. On February 8, 1865, he delivered his first lecture on pea experiments to Brunn Natural Science Society. His paper, Experiments on Plant Hybridization, proposed some basic genetics principles. However, his work was unattended and unappreciated up to 1900. It was only in the beginning of 20th century that Hugo de Vries, Karl Correns, and Erich von Schekmark were working on various flowering plants independently drew the conclusions just like Mendel. It was William Bettison who confirmed Mendel's work by series of hybridization experiments. Now let us see Mendel's selection of the experimental plant. Mendel was very careful in selecting his experimental plant. He found the plants of family Leguminosae such as peas and beans, most suitable materials for his experiments. In his experimental plants, it required constant variations, and also the hybrids of the plants must be protected from the influence of foreign pollen grains. Now let us see some reasons behind why pea plants were selected by Mendel. The first reason is pea plants are easy to culture in open grounds and also in pots. Next, pea plants showed short growth period and life cycle. Pea plants showed peculiar structure of flowers in which there was self-pollination. However, artificial cross-pollination was carried out in order to bring about fertile hybrids. Mendel chose this plant because they showed contrasting heritable traits. For example, the height of the plant, either it is tall or short. Pea plants also showed large number of offsprings, which helped Mendel in propagating more and more pea plants. So these are the reasons why Mendel chose pea plants for his experiment. Now let us learn about Mendel's procedure, how he brought about his experiment. So Mendel cross-pollinated the flowers by hand by simply opening the flower buds. He then removed their pollen-producing stamens, as you can see in the picture, with the help of a scissor. This operation of removal of anthers is called as emasculation. He then took pollen at the dehiscence stage from a different plant and used that plant as male parent. With the help of a brush, pollens were dusted on the feathery stigma of the emasculated flower. He then made sure that the stigma of the flower was protected against any foreign pollen with the help of a covering bag, as you can see here in this diagram. After hybridization, the pollen should be categorized. Seeds are to be planted or sowed in the succeeding season, and the plants are raised to maturity. At the beginning, Mendel established through breeding varieties in which plants produced progeny like themselves and allowed to self-fertilize. Later on, when two varieties differing in one or two traits were crossed, a hybrid progeny was obtained. In his experiment, Mendel chose 7 contrasting traits, such as the seed form was either round or wrinkled. The cotyledons were yellow or green. The color of the flower was white or else purple in color. The form of the pod was full or constricted. The color of the pod was yellow or green. He also took stem as one of the contrasting traits, in which he chose the place of flower that was either eggshell or terminal. He also chose the size of the plant as one of his characteristic traits, that was either tall or short. Now let us learn about Mendel's experimental observations and results. The hybrids of F1 revealed that plants contain the character of only one parent, and none of them displayed any intermediate character of both parents. This can be explained as follows. A phenotype of purple-colored flower was crossed with a phenotype with white parent flower. The result of this were all purple-colored flower in F1 generation. The characters which were transmitted almost unchanged in hybridization process were called as dominant characters, and those which were suppressed in the process were called as recessive characters by Mendel. This can be shown as follows. So we take the previous cross and point out this particular flower, that is purple, as dominant trait or dominant character, because in F1 generation, all the offsprings were seen showing purple colored flowers. On the other hand, white colored flower was a recessive one because this particular character was suppressed in F1 generation. He further noticed that it was perfectly immaterial whether the dominant characters belonged to seed bearer or to the pollen bearer parent. The form of the hybrid remained identical in either cases. In the first case, you can see here the purple color flower is representing the male parent and the white colored flower is representing the female parent. However, in the offsprings, all of the offspring showed purple colored flowers. In the second case, the purple color flower was represented as female parent and the white colored flower was represented as male parent. In this case as well, all the offspring showed purple color flowers, which was the dominant character. Mendel's observations of F2 progeny further revealed that the fact that the recessive character, which was hidden in F1 generation or F1 hybrids, reappeared in the F2 offsprings and expressed itself at the average proportion of 3:1. So here is the explanation of monohybrid cross, in which selfing was carried out in F1 offsprings, and in the F2 generation, 75% of the offspring showed purple colored flowers and 25% of the offspring showed white colored flower in the ratio 3:1. Further, when the recessive hybrids of F2 were self-pollinated, they always produce F3 offsprings with only recessive characters. As you can see here, these are the F2 generation offsprings which underwent selfing, and it produced 100% of recessive traits offsprings in F3 generation. The remaining 75% from the F2 generation showed in F3 generation about 25% hybrids with only dominant characters. And on the other hand, the remaining F3 generation were having 50% hybrids which yielded dominant and recessive characters in the ratio 3:1. On the basis of the results of his experiment, Mendel recognized the phenomenon of dominance and formulated the following two laws: the law of segregation and the law of independent assortment. These are my references. Thank you.